things have not gotten worse, even though we have this teaching in the lecture hall and the like. So I think we're lucky.
Oh, so now we're recording, I see.
Yes, so I'm going to introduce everybody in. See some friends online. Ooh, lots of
Very good.
people.
Okay.
Hello, hello.
This is the fun part. Okay.
Okay.
Yeah.
Yeah, so maybe you want to put the slide on, Gilad?
Yeah, I'm gonna do that.
Yeah, so maybe you want to put the slide on, Gilad?
Yeah, I'm gonna do that. Uh-oh. That's my.
Hopefully the sun will come up and I'll have a little more light and it won't be so dark for me as we get going.
No, we see you quite well, Susan. That's
Oh,
okay.
good.
You will.
Okay. Maybe I'm just not awake yet. I don't see myself very well.
So let's see who's online there. I saw Dave before. And Bob Matthews. And lots of other friends, Michael Hack. Sheena. Pink Chen. Yeah, lots of people. So we have a couple of more minutes. Hi, Sheena.
Hi, how are you?
Fine, and you?
I'm good, thank you. Surviving.
Good. Good
Yeah,
to
be nice to see the inside of my lab at some point.
I haven't been back.
see you, love.
Nice to, you know, so the group are working about half time, but we're not allowed in because we're not useful.
Yes, we're not essential, Sheena, is what we've been all found out.
So I'm having an intimate relationship with my laptop right now, and it's kind of sometimes feels like divorce would be appropriate. How are you, Susan?
Well, we are probably the only crazy ones who opened early everything completely freely, and now we're in a second lockdown.
Are you completely locked down, Gilad?
Yeah, well, completely is a relative term.
Look where he is, Sheena. He's in his office.
I'm in my office.
Problem was that Michael Levitt was sort of conceptually correct, but he had the numbers wrong.
Sorry.
But it was conceptually correct. I think there's a lot of stuff, I mean, probably for different reasons, but I've been looking at this. It really is true that the end game is very hard to predict, and it can sort of almost miraculously disappear. That once you go past the exponential phase, the cutoff point is very unpredictable.
That's what Donald says.
What?
It miraculously disappeared.
Yeah, and that's unfortunately what Trump says. See, sometimes even stupid people are right, you know, occasionally.
I'm still waiting for it to miraculously disappear. But
Yeah, no, I think, but it will one day miraculously disappear, but it will not be a miracle. It will be the result of chain reactions in a spatially disordered system.
People are
You know, you know. but you will be very hard to know when that point comes. Oh well, sorry. There's actually an article pointing this out. There are many articles on this, but one is in PNAS last week. So
okay.
you can look at some of the math. I can't vouch for the math of that paper. I did it a different way, but yeah.
Sounds like a good topic for a seminar.
Yeah, journal
Not by
club.
me. Not
That's
by
teacher
me.
one.
You can only get in trouble with this topic, I think.
Yeah. Maybe we can get Mike to talk about this in the webinar here.
Yeah.
Oh, I bet he would. I bet he would.
Yeah. Right. Pandemic folding and dynamics. All right. I guess we can start. It's 2 minutes after the hour. So hello, everyone. Welcome to the protein folding and dynamics webinar. This is actually our 12th lecture in this webinar. We started almost exactly 5 months ago. And it's been quite a ride. In the beginning, we thought that we will just do that for maybe 2 or 3 months. But it seems like it's going to continue quite a while. We are already booked way into 2021, and we will continue with that as long as there is a demand. And there's still a very robust audience, as you can see. So we are quite happy with that. So let me just, before introducing the lecture of today, let me just say a few words about the way we handle the lecture itself. So I will ask you all to mute yourself. I think that most people are now already mute. So please stay mute during the talk. But after the talk ends, we will have ample time for questions and answers. And so if you have a question, please go to the chat sidebar and just write that, either write your question or write that you have a question. And in the end of the talk, I will ask you to mute yourself one by one and ask the question. With that, I think I finished all the preliminaries. I would just like to mention that in 2 weeks' time, we have a lecture by Henry Chapman, who will talk about serial femtosecond crystallography. This is a very interesting topic too, and I welcome you all for this talk in 2 weeks. So I would like to turn to our lecturer of today, Professor Susan Marqusee from the University of California at Berkeley. Susan is a professor of biochemistry, biophysics, and structural biology at Berkeley. And she is also a director of the Berkeley branch of the California Institute for Quantitative Biosciences. Susan did her PhD and MD at Stanford. She worked in the lab of the legendary Buzz Baldwin, where she did seminal work already as a PhD student on helical propensities of amino acids. She then postdoced with Bob Sauer at MIT. And in fact, Bob has on his website this alumni page. And if you go to that page, you will see a lovely photo of Susan and her husband and her baby daughter. I guess that was around 1990 or maybe 91 or something. Susan then joined Berkeley as a faculty member and has been there ever since. Over the years, she did very prominent work on protein folding, and she's won many awards for that work, including, among others, the Margaret Oakley Dayoff Award of the Biophysical Society, more recently, the Dorothy Crawford Hodgkin Award of the same society. She won the Stanford Medicine Alumni Award in 2017. And in 2016, she was also elected as a member of the National Academy of Sciences in the US. Her work revolves around various topics of protein folding dynamics, understanding the energy landscape of folding, and most interestingly, understanding various factors that may affect the energy landscape, including cellular factors and environmental factors, of various sorts. She uses a large and rich ensemble of techniques in order to study protein folding, which go all the way from mass spectroscopy to single molecule force spectroscopy. And I'm sure we will hear about some of these techniques today. So without further ado, I will give the podium to Susan to talk about modulation of the energy landscape by cellular factors. So please, Susan.
Okay, thank you, Gilad, and thank you, I think, for inviting me to speak here. I have to say it's a little bit odd. Let me first share my screen, hold on. All right, so first of all, thank you, Gilad, for inviting me. And this was the first announcement Gilad sent out with this title. It was the middle of the night for me. I happened to look at my phone, a bad habit, and panicked. I had 1 week to learn everything I could do about uphill relaxation and thermodynamically equidistant temperature quenches. But Gilad sent me the paper, so that helped me out. And in the end, I decided I would go ahead and give the lecture that I was going to give in the beginning. I'm a bit tired. It's quite early in the morning here. And last night, as I finished up preparing my slides, we had a blackout and no power in the mountains where I am now trying to avoid, or I'm trying to spend time either in our house in the mountains or at home, wherever the air quality is better. So luckily at 1 in the morning, the power came back. So here I am, and we'll see how this goes. Okay, so everybody can see the right thing, the right screen? Yeah, okay. Let me just move some of you guys around so I can see. Just 1 second. I move that to, shoot. Let's put it down there. Okay, so I think we'll start where everybody in this audience always starts, which is, it's all about the energy landscape, and that's where biology comes from. We have the ground state, which under physiological conditions is going to be the native structure of the protein usually, and all of the fluctuations and their dynamics that allow a protein to function and turn over and everything else. So the theoreticians can see everything, computational biologists, theoreticians, they can see this energy landscape. I'm an experimentalist, and as an experimentalist, we really usually only have 2 things on our plate that we can, that we can look at. And so usually we can look at the energetics between the states. Here I showed the folded and unfolded, and the kinetics between them. And then we map them back on, and if we're to this energy landscape, and if we're lucky or clever, we can actually see the structures of these different conformations. So I think I don't have to tell this audience again that this energy landscape is not only decided for a protein, it depends very much on the sequence of this topology and on the environment. And we all know that if you have, add urea, the protein will unfold, add temperature, the protein will unfold. And usually we will assume that, if the rates change and the folding and unfolding for this 2-state protein, we'll assume that we've affected this barrier height as well. We can also change the sequence, and those things will change as well. So we spent a lot of time in my lab over the last decades characterizing and developing technologies to probe this energy landscape in vitro. And we use the changes in the sequence and changes in the environment to do that probe. We may use urea, we may use mutants to actually probe and understand. And that's actually not the topic of what I'm going to talk about today, but I just want to let you know how much we and others, and many in this audience, have learned over the years about the in vitro energy landscape of a protein. Sorry, it's going to take me a minute to get the cursors all in the right place. Okay, oops. So over the years, we've spent a lot of time, as many of you know, in my lab studying a particular family of protein called ribonuclease H. And at this point, we know quite a bit about this protein from many different experiments, as Gilad pointed out in the introduction. We know much more than just its folding rate and unfolding rate. We know by using some nice hydrogen exchange that was originally developed by Walter Englander, we know the population of many and structures of many conformations along this energy landscape. We even know the folding pathway in terms of which regions of secondary structure form first and what controls the different rates of that. We do these experiments again by manipulating the environment by adding urea or guanidine to study them. Similarly, we can change the sequence. We've made lots of single sequence, and we've even created the ancestral tree for this family of proteins, where we can reconstruct and synthesize sequences that are the presumptive ancestors between two extant homologs of RNase H, and study their folding and energy landscape at a very detailed way and figure out what pressures in the landscape. And what we see is that landscape I showed you before holds for almost every one of, or all of these ancestors that we've studied to date. So all of the RNase H's that we've studied to date, we can simplify in this two-dimensional projection as having an unfolded state rapidly folding to a partially folded, marginally stable intermediate that then goes over the folding barrier to the native state of the protein. And this usually for most homologs is a millisecond pre-equilibrium to the intermediate and then a slower seconds folding. We know the region of this intermediate, and it's this blue region hydrogen exchange has told you. The reason I want to show you is because I'm going to be using a particular mutant. We know enough about the energy landscape of this protein that we can make site-specific mutants and change the folding kinetics and perhaps the energy landscape in predictive ways. So if we put a charged residue, a spartate, in the middle of the hydrophobic core of this protein, we destabilize this intermediate, so it is now higher than U, and the protein folds in an apparent two-state behavior. And I'm going to use that protein later on. So the goal of much of the work I'm going to talk about today is trying to be able to figure out how the cellular environment and the specific factors in the cell might modulate this energy landscape of a protein. And that's technically much more difficult because the cellular environment is not really conducive to the types of biophysical studies that we use. The cell, as you know, is complex and crowded. The protein is interacting with specific partners. How do we actually obtain, homogeneous biophysical quantities of these proteins bound to their factors? When we study them in these crowded conditions, they tend to aggregate, etc. So there's been a lot of biochemists and cell biologists who have studied the energy landscape, or at least the folding pathway and how molecular machines interact with that folding pathway in the cell using biochemistry and cell biology techniques. And what we want to provide to that is a link between those in vitro biophysical studies and the cell. So we want to sort of reconstitute this piece by piece to understand how these factors modulate the energy landscape under the in vitro conditions. Are there big changes or not? And extrapolate them, and hopefully we'll come and meet these biochemists and cell biologists to understand the whole pathway. So why do I think that the energy landscape would even change under environmental conditions? How much of a change could there be when they do that? And so before I go ahead and start with the details of today's lecture, I want to just show you a result that's maybe almost over a decade. Well, no, a technique that we developed over a decade, but the results are about 5 years old, looking at how we can actually probe the transition state or the trajectory of protein folds to give you a hint not only of that these things small changes can dramatically change the energy landscape, but also to introduce you to a technique I'm going to use later on. And so many of you know, 15 years ago, my colleague Carlos Bustamante and I developed a pretty simple technique. We heard a lot about it from Matthias Reif a few weeks ago, single molecule optical trap spectroscopy, where one can monitor the unfolding and refolding of a single molecule in the optical trap as a function of force. So for those of you, just a quick reminder, in this experiment, we have 2 polystyrene or plastic beads with a protein tethered using disulfide chemistry to 2 long pieces of, this is not to scale obviously, to 2 long pieces of DNA that serve as a spacer between the beads. One bead in our system is held in an optical trap, the other bead on the tip of a micropipette, and we move either the optical trap or the micropipette. We can move them apart and apply force and monitor the force and the distance between them. And I think you all know, as you pull and stretch on a protein, eventually the protein will not maintain its native structure, and it will unfold to an unfolded state that is more extended and therefore favored under force. So let's talk about that a little bit more quantitatively. This is the energy, Gilad, I don't have a clock on my screen, so if things get bad at the end, let me know. So this is the energy landscape of the protein, say under zero force, and we have the folded state populated. We add force and we bias the unfolded state because it has a further distance or end-to-end distance in the protein. We can quantitate how much we've biased the unfolded state over the folded state as a function of force simply by force times the distance between the endpoints in the folded state and the unfolded state. So as long as the unfolded state has a larger distance, it will be biased by adding force. Just like we can quantitate the energetics, we can quantitate the kinetics by looking again at how much we've changed the energy of the transition state, again by its distance to the transition state. So the rates are going to be modulated by a FDX term, where now this dx is the distance to the transition state. So why am I telling you all that? Because this means that if I plot the log of the rates or the height of this barrier as a function of force in these experiments in a very simple model, which is fine for what I'm going to show you today, the slope of that gives me this distance to the transition state. It's a characterization or a number that gives me some characterization about the transition state of this unfolding or folding trajectory. That should look very similar to many of you who have seen chevron plots, because in a chevron plot, we do the same thing. We have an environmental perturbant, in this case, urea or guanidine. We plot the height of that barrier and we see a chevron, and this slope is the M dagger of the unfolded is again, tells you something about the distance to the, or tells you something about the placement of the transition state and is a characteristic of the folding trajectory. So I can characterize the folding trajectory by either characterizing the distance to the transition state or the M value to the transition state. And if the folding landscape doesn't change very much along these environmental perturbants, be they force or urea, then I'm going to have a single distance to the transition state and a single M value, and I will characterize that energy landscape. So what we sought out to do is combine these two environmental perturbants and ask whether there was a single M value and a single X dagger value to characterize the transition state, or under certain, another way to say it is, would the M value change with force, or does the X dagger value change with urea, such that we maybe have different characteristics of the transition state or different transition states under different environmental conditions. So we did that experiment on a simple protein, SRC-SH3. And what you can see here is simply, first we can just look at the red, which is this distance to the transition state or the slope, and this is not a straight line. This is best fit by a sum of two straight lines, suggesting that there are likely two, the simplest suggestion is that there are two transition states that the protein covers, one dominating at low force and one dominating at high force here. If that's true, then they should have very different M values. So we also did the urea dependence as a function of force, and here I'm just plotting the value at 1 molar urea because this is a three-dimensional plot, and this distance here is going to be the M value. And what you can see is the M value is very different at low force than it is at high force, suggesting that the transition state cannot, or the folding trajectory cannot be characterized as a function of force in urea here as a single barrier, but more that's changing its height, but rather that the protein, the majority of the flux is crossing a different barrier at low force here than it is at high force. So again, I can just plot that flux as a function of force. And so here what you can see is, yes, under any condition, we just measure a rate. For a two-state protein, we would just measure a rate of folding. And at low force, that rate would be dominated by this pathway I'm terming S1. And at high force, it's going to be dominated by this pathway S2. But here somewhere, I don't know, 23 piconewtons or something, we actually measure a single rate, but it's 50% of the flux is going over each, 50% of the flux is going over each barrier. So even though we can't detect it, it's important to remember that there are, I don't know why my mouse keeps disappearing, that there are two different hills that the protein is climbing here. Now I'm going to change the environment, say at this 23 piconewtons, and I'm going to add 1 molar urea. Suddenly I've changed, by just adding 1 molar urea, I've changed it so I have, so that I have 60% of the protein going through one barrier and 40% going through the other. So just adding 1 molar urea will change the flux and the trajectory that the protein's been folding here. Similarly, I can make a small change in the sequence, just 1 amino acid change, and I can also change the flux. So just concentrate here on this single mutation T50A. This is the wild type or reference sequence. And now when I make that mutation, I don't change the properties of this S2, but I do change the height and the barrier of S1, suggesting that T50A is involved in the energy barrier, that site for only this, that they're discrete regions of the protein that are restraining or responsible for holding the protein together and the transition state for the unfolding trajectory under that force. So that has a lot of important aspects for unfoldases and what forces the protein is undergoing in the cell, but that's not what I want to cover today. For the main point of this, I really just want you to notice that very small changes in the environment can have very big changes in the energy landscape of a protein. We all knew that for simple mutations, but it can be quite complex. And so with that, I want to get to the point of today's lecture, and we will come back to this experiment. So today, I want to talk about 2 different modulations of the energy landscape by cellular factors. First, I'd like to talk about the ribosome and co-translational folding, or the birth of the protein. And then I'm going to talk about ubiquitination and the death of the protein. So I know if you're like me, you're probably checking your email and getting texts at the same time, or just get Zoom fade out. So you don't need to know anything about the first half when I talk about the second half. So if you space out, just join in and you won't lose anything. Okay, so let's talk about co-translational folding. And so now when you think about co-translational folding, this is really an extreme example where we're changing both the sequence and the environment. In bacteria translation, we can think about it occurring at about 10 amino acids per second, which means that it takes quite a bit of time for a protein to be translated. Much more time than it takes, as we already know, for the dynamics and conformational sampling of small domains or regions of secondary structure, et cetera. So during translation, all proteins are going to have an opportunity to sample conformations and fold while on the ribosome. And the energy landscape is going to change as a function of time as the chain grows in length. So lots of people, again, in biochemists and cell biologists have been studying this process. I'm by no means the first. Some of them are on this audience. And there are some really beautiful cellular ways to look biochemically at the factors that might be involved. I think Lila's on the line, et cetera. And there's some very detailed biophysical studies to look in sort of on the ribosome in vitro translation and by NMR, single molecule trap spectroscopy, et cetera. But I'm going to talk a little bit about some more general techniques that allow us to probe different proteins and different sequences a little easier so that we can relate back to the in vitro folding pathway. So why do we care? This is a student of mine, Sean Costello, made this plot for me to think about why do we really care what happens during translation? Because the protein has plenty of time in its life maybe to do other things and refold, et cetera. So what I'm taking here is a bunch of 2-state proteins that we all know well, and I'm plotting, or Sean has plotted, the folding time over and against the, I think that says the unfolding time in seconds. And so what you can see, if we assume, let's just do a back of the envelope calculation, that it takes about 30 seconds to translate 300 amino acids. So what that's going to tell us is every domain to the right of this purple line could fold during translation, but not unfold during translation. So all of these will fold as it has the opportunity at least to fold during translation, and that will be what it is during the whole translation process. Then if I make the assumption that 45 minutes, just taking yeast data, is the average lifetime of a protein, everything to the right of this orange line would only fold during translation, and that's the only time it's going to fold in its lifetime. So what this says, what happens on the ribosome could have a very big, and will have a very big effect of what happens in the cellular lifetime of the protein. So it's pretty important for us to understand how the different features of the translation apparatus and the translation process affect that folding process. So the first thing we wanted to do in this deconstructive approach is just ask, what about the ribosome itself? When you're translating, you're attached to this large electrostatically charged protein nucleic acid complex how does that, it does have a large potential, affect the stability and the energetics and dynamics. So to do that, we turn to stalled ribosome nascent chain. So we can make a protein, a nascent chain, attached to the ribosome, and you can isolate it by using this sequence called SEC-M. It's a stall sequence. It will stall the polypeptide wherever you put this into the in the ribosome. Sorry, it's a bit early in the morning for me. And you can purify this ribosome nascent chain. Well, that's fine. And I think John Christodoulou does beautiful NMR by labeling the chain, et cetera. So how are we going to study the simple energetics of this protein RNA complex? We can't exactly put this in the CD or the fluorimeter and easily study it, since most of the protein and nucleic acid comes from the ribosome. So to do this, we turned to a gel-based technique that was developed in my lab now in 2005 by Ji-Wook Park when he was in my lab, called pulse proteolysis. So in this case, we make a stalled nascent chain and we label the protein with a fluorophore during its in vitro translation. So we can follow in a gel the protein of interest, and all of the other proteins in the ribosome will be blind in our gel. We then set up a condition where we have proteins that are in equilibrium, the folded and unfolded state, just like we do in a CD melt, et cetera. And we take advantage of the fact that a protease will much more quickly degrade the unfolded proteins than it will the folded proteins. So we give it a pulse of the protease, we degrade all of the unfolded proteins, and we simply run a gel and ask how many proteins are left, and those were the folded proteins. We don't do this pulse for long enough that the folded proteins can re-equilibrate to the unfolded state. So we know how many proteins we started with, we know how many we do after the pulse, and we can just plot that and look at it on a gel. So what you see here, we use thermolysin, which is quite robust this is a protein off the ribosome, I should tell you, quite robust to urea. And you can just see with that protein RNase H, we can plot the intensity of this native band and plot out the equilibrium unfolding of RNase H. Now, it turns out that the ribosome itself is stable up to about 2.5 or 3 molar urea. So we can do this experiment as long as I don't have to take the experiment higher to that. So to do that, we used a variant of RNase H, which would have its stability in the range, and said, could we, using in vitro translated protein, determine its stability, and then do it again in ribosome nascent chains. And actually, Sophie Jackson had already used this technique on in vitro translated proteins, so we had a good idea that it would work. So here's the stability curve, or the unfolding curve for this 2-state mutant of RNase H, I53D, off the ribosome and on the ribosome. And you can see for this protein that being on the ribosome destabilizes the protein. Moreover, one can ask, does this have anything to do with how far away the folded domain is, or the domain that's folding from the ribosome itself? So we studied a series of proteins where the target protein was, in this case, RNase H I53D, was separated from the SEC-M and the PTC, or the ribosome translating site, by either 10, 20, or 30 amino acids using a flexible GS linker. And what you can see above is, as we get further and further away, the modulation by the ribosome goes away. So by the time we're at 30, a linker of 30, it's back to its stability that it was off the ribosome. So we've done this on many proteins to date. We've seen always pretty much the same result, that the ribosome destabilizes the protein, and it does so in a distance-dependent fashion. So we get the simplest energetic difference. This is a 2-state protein. Can we learn something about the rates of folding and unfolding? And so Madeline Jensen recently in my lab took this approach, and instead of studying the equilibrium population, she did an unfolding experiment where she diluted the protein, or the ribosome nascent chain, away from denaturin into folding conditions. And she just used pulse proteolysis to get a snapshot as a function of time of how much folded protein there was. And this is what you can see here. This is off the ribosome, and these are on the ribosome. So we can simply calculate those rates of unfolding. And again, you see off the ribosome and on the ribosome. And first of all, what you see is that it unfolds faster when it's on the ribosome. So it's destabilized and it's unfolding faster. And in fact, that difference in stability completely accounted for by the change in the unfolding rate is completely accounted for by the increase. Let me start that again. The change in stability we see in equilibrium conditions is completely accounted for by the change in the unfolding rate. In other words, we can then make the assumption that the folding rate is unchanged on the ribosome. For this protein, it appears that the unfolding rate is accelerated. So this is pretty interesting. We've had the protein is destabilized when it's close to the ribosome and it unfolds faster. And it's just physical chemistry, but it's also could be of benefit to the system because as you're translating, you have incomplete sequence exposed out of the ribosome. If it samples a structure, it could well be the wrong structure, and you want to easily get out of that structure so you can form the right structure as the protein continues translating. So having this increased unfolding rate and decreased stability could be a good thing to do, teleologically speaking. Now, the other thing I want you to notice here is the slope of these lines. That's the value. That tells you, again, something about the transition state for unfolding, and these are parallel. So this suggests that actually this protein is folding through the same basic trajectory on and off the ribosome based on this. And so I'm not telling you in the order we actually did this, but I've already showed you an experiment where we can probe in detail by doing that chemomechanical unfolding and looking at whether or not the transition states are the same for the protein. So we went back to that protein SRC-SH3, Emily Gwynn in my lab, and using a technique that Christian Kaiser had developed, we could, in the optical tweezers, capture a ribosome nascent chain and do that same experiment that we had already done by following unfolding as a function of force and urea for the SH3 domain on and off the ribosome. Although this is the same protein we studied before, we're actually pulling from a different position in the protein, so we had to repeat both experiments. So here's the data for off the ribosome, and in this case, pulling from the N and C terminus like this, we see a straight line consistent with only one unfolding transition state and an M value of about 0.3. This M value is the same as the M value we see in bulk, suggesting that the protein on the trap and in bulk is unfolding through the same trajectory. What about when we look at it as a ribosome nascent chain? Well, we saw the same results. Here again, we see that the unfolding M value is the same as it is both off the ribosome and in bulk, suggesting that the same pathway or trajectory is taken both on and off the ribosome for this small protein. This has also been a result seen by Jane Clark and Robert Best for another small beta sheet-like protein, suggesting maybe that for these small domains that the energy landscape or the fluctuations and their dynamics are controlled by the same general principles, both on and off the ribosome, which would be very nice for moving forward in trying to understand the modulations as we go from these more simple in vitro experiments to more complex. Okay. So that's our experiments looking at the ribosome, but that's not really co-translational folding, right? In co-translational folding, you've got this non-equilibrium process where the protein is always being, kinetically it's translating. There's people like Patricia Clark, Ed O'Brien, lots of people wondering how that rate of translation is affecting the folding process, et cetera. So how can we study this in real time in a sort of robust way to do that? And we thought about this quite a lot, and a lot of people who want to study anything complicated in the cell, they go to GFP because GFP fluoresces and you can study it in those complex situations. But of course, as we know, GFP, the rate limiting step, is the maturation of the chromophore, and I'm not interested in studying the maturation of the chromophore. So instead, we turn to another genetically encoded fluorescent tag that a lot of people study, and that's HaloTag. So HaloTag, for those of you who don't know, it's a modified dehalogenase that Promega has mutated and engineered, so it stops at the covalent intermediate. That means that you can give it a synthetic dye, a halogenated dye, it will react and covalently attach to the protein, and the protein will now be fluorescent. So we wondered how fast that dye binds, which binds in the active site, so presumably it only binds to the folded protein. And we know that the ligand binds quite fast. So if under our conditions of the amount of ligand we add, that the on rate of the ligand is much faster than folding, we could use ligand binding as a probe to tell us when the protein is folded. So let me, the one thing about this, of course, I can't see anybody, is that I can't see your faces to see if you're following, but I will try to, I think that was a bit complicated, so I'll try to repeat that. So here, what we're doing is we're asking just that. We're gonna take the protein in the unfolded state, we're gonna dilute it into urea for HaloTag in the presence of this dye. Now, unlike GFP, this dye is gonna be fluorescent whether or not it's bound to the protein. So we're not gonna just use fluorescence to study binding, we're gonna use the size of a fluorophore or fluorescence polarization. And under our conditions, the dye binding rate is sub-milliseconds, and it turns out, as you can see, the protein folds much, much slower. So here I'm just monitoring that folding of the protein as the dye gets bound to it as a function of time. I extrapolate the folding rate of the protein, and if I compare that to what I see in CD, they are very similar. So the fast rate of binding is allowing us to use a ligand. The fast rate of binding allows us to use ligand binding or fluorescent polarization as a proxy for protein folding. So once I have this proxy for protein folding, I can do this in really complicated conditions. In other words, in the presence of the ribosome and all the translation factors, et cetera, because I'm just following the binding of this dye onto the protein of interest. So we did this same experiment in an in vitro translation system. Here, what we're doing, this blue line, is we're just adding a purified recombinant, it's called Pure Express, in vitro transcription translation system, giving it the DNA for the protein of interest in the presence of the ligand, and just watching in a fluorimeter over time what happens. And what you could see is there's some lag time, presumably for the protein to be, the DNA to be transcribed and the protein translated, and then we see a folding of the protein. Importantly, that if we don't give it the plasmid or the DNA for HaloTag, but for DHFR, we don't see binding of this dye. So we're really seeing folding of HaloTag as a function of time. But we aren't really necessarily seeing folding. We are just seeing how long it takes to transcribe, translate, and fold the protein. So I don't know what the rate limiting step here. Is this protein synthesis? Is it every time the protein gets translated, it folds? Or am I actually monitoring the folding of the protein? Notice this is very, very slow time scales. So to do that, we decided we needed to know how fast the protein was being made and seeing if the protein is being made slower than this protein is folding. And so to do that, we simply incorporated a fluorescent dye into the protein, did this in vitro translation mixture, and then just ran a gel and looked to see how much protein was translated or produced full-length protein as a function of time. And that's what's shown here in the red line. So this tells you the accumulation of folded protein. Oh, sorry, erase that. This tells you the accumulation of protein product, and the blue tells you the time course of folded protein. So these are complicated non-equilibrium experiments, and so we sought out Ed O'Brien, who's thinking about the same things. And Ed first solved for the rate of translation as a function of time, and that changes with time as we run out of factors, et cetera. And here you can see our in vitro translation mixture is pretty standard. We're getting about 2 amino acids a second at its optimum. And then on top of that, we were able to fit the folding rate of the protein. And we got pretty similar folding rates to what we had seen in the in vitro refolding experiment. So the rate of folding doesn't seem to change very much. And of course, it's really slow. The protein is doing its final folding off the ribosome. This is a very slow process. So we thought, okay, this is very interesting. We should really characterize the folding pathway of this protein that had never been studied before in vitro refolding and ask, is it exactly the same or is it different? And we found some important differences. First of all, as I told you, I'm just going to summarize for time here. The overall rate of folding is pretty much the same in refolding when you dilute from denatured. I'm going to always show you this marker as a refolding, and I'll show you a picture of the ribosome when I'm talking about in vitro translation, because they are both in vitro. So the rates are the same, but what we found out is that when in low urea, that there's aggregation occurs when we do refolding experiments. That is, you can see there's this fast phase and then the thing just falls out of solution and the signal dies. So we see aggregation. We have no evidence for the same aggregation during in vitro translation. So folding is much more efficient during translation, and that there's some kind of process that occurs before this aggregation occurs. So that made us make the following hypothesis. We thought, what does this aggregation prone intermediate look like? And perhaps it doesn't form during translation, and therefore co-translational folding is taking a different product that is more efficient than it does during the refolding process. So that was our hypothesis. You'll see in a minute that it does come out to be true, but we asked, how could we find that out? Well, we thought we could look at what does this aggregation prone intermediate looks like in refolding, because we had done that before using a technique that many of you are aware of called pulse labeling hydrogen exchange. So in this case, it was originally developed by Buzz Baldwin and Walter Englander using NMR, but now we're using mass spec to analyze it. So in this experiment, we take the protein, it's completely deuterated. We let the protein fold for a while and we ask, okay, whoever's unfolded at this time, exchange all your deuterons for hydrogens, and then we will be left with a fingerprint of what is folded, and we can quench the reaction, digest it up to peptides, and look to see where these deuterated protons were. So this is actually a little bit of a difficult experiment on such a slow, large protein like HaloTag, but I'm just going to summarize the results for you and not the details of how we did it. So when you do this experiment, what you find out is peptides in this red region are folded in this early time point, and peptides coming from the white regions are protected on a much slower time course. So this maps out an early folding intermediate, the red region. And two things that are interesting here is, one, you can see like perhaps where this helix is here, that there must be a hydrophobic surface that's exposed, that's very easy to aggregate. So we could, like most intermediates, assume that this is an aggregation prone intermediate. Moreover, we were particularly interested that there are regions, and this is a simplified one-dimensional view, regions from both the N terminus and the C terminus that comprise this folding intermediate. So one could imagine as you're translating, when you don't have both the N and the C terminus exposed, that you would rapidly compact to some other type of intermediate and bypass perhaps the folding of this intermediate that's aggregation prone. So that was our hypothesis, only we didn't know how to test it. Because I think I do now, but at the time I don't really know how to do this pulse labeling hydrogen exchange experiment on ribosome nascent chains. So instead, after discussions in the lunchroom with Andy Martin, who has the lab next to me, we realized we're really testing a hypothesis. We don't need to look at every amide protein in this study. We can just ask, if I put a modifiable side chain, say a cysteine, in a particular region of the protein, and I ask at what time during folding does that cysteine become unavailable for modification from a solvent probe, and I'll show you that in a second. So in other words, we're going to do the exact same experiment, but instead of looking at amide protons, we're going to look at the availability of a certain residue for modification by the solvent. So we can do that experiment either by refolding or by in vitro translation. So we can take the protein, we start it folding either way, and after some amount of time, we just label that cysteine or attempt to label it with a fluorescein maleimide. If that cysteine is not protected by folding, it will be rapidly labeled. And then we can go ahead and run the protein on a gel and say how much of the protein is fluorescent and how much over time, and get an idea of when that particular cysteine becomes protected. So we did this experiment we call pulse cysteine labeling, and the wild type has several cysteines. So we just did it on the wild type first. And so what you can see is there's two cysteines here, and what I'm showing you in the black is the refolding. One of the cysteines is we predict would be exposed in the intermediate and the other one protected. So what you can see here is exactly that. Rapidly, the black line, you see about half of the signal gets protected quickly, presumably in the folding of this intermediate, and then under a slow time course that we see of the folding rate, we see the other one protected. That is not what we see when we do the in vitro translation experiment. We make protein and then we stop synthesis with an antibiotic, and what you can see is that there's a slow single exponential phase. I'll show you in a more convincing plot in a second, as the burial or protection of the cysteine residue, of these cysteine residues. So the folding looks different co-translational and in vitro translation and in refolding. I think I can show you this more convincingly by taking out and only looking at one single cysteine. So I'm going to show you the example from one cysteine. This is a cysteine that we should be buried based on our model for the structure of the intermediate. And what you can see is right after we start folding, it immediately becomes protected to its final equilibrium protection level, exactly as we would expect it. This is where it starts unfolded, and then it becomes protected. Now, when we do that experiment in co-translation using the in vitro translation mix, what you see is it does not get protected very quickly, but it gets folded or protected on a very slow time scale, consistent with the overall folding of the protein. So we've looked at several cysteines, not that many, and we can see that our hypothesis seems to be holding true, and that is that the folding trajectory, or this intermediate that forms an in vitro refolding, does not form during the co-translational process. So what appears to happen is translation reroutes HaloTag's folding trajectory, bypassing this intermediate that seems to be aggregation prone. We don't know what intermediate is forming during the translation process, and that's what we're working on now. We don't know how much the effect of the order of the synthesis, that the N terminus comes out before the C terminus, plays a role. And Natalie in my lab is busy making every circular permutant of this protein to examine how much the topology is affecting that. And Sean Costello in my lab is developing technologies to be able to actually monitor the folding trajectory in the co-translational experiment. Okay, so in the last bit, that took a lot longer than I thought. I will tell you this story if that's okay, which is, I will do much faster, which is a new work on ubiquitination of the protein. Okay, so just to get everybody up to speed, as you know, ubiquitination is a protein, 8.5 around kilodalton protein that gets appended on lysines of proteins and sends them off to different cellular fates. And the actual ubiquitination process is a complex regulated cascade of enzymes called E1s, E2s, and E3s, which put ubiquitin on lysines of proteins, mostly lysines. And since ubiquitin itself has lysines, it then gets added to form chains of ubiquitins. The most notable or famous cellular fate of ubiquitinated proteins is sending it to the proteasome for degradation. And this is work for, I think, Kobe, I saw you're on the talk here. This is a paper from figure from his paper. And so in order to be degraded by the 26S proteasome, two things have to be true. You have to be ubiquitinated and recognized by the proteasome or ubiquitin receptor. And you have to have, if you're a folded protein, you have to have a disordered or unstructured region that allows for engagement into the 26S proteasome and for pulling, unfolding, and eventual degradation of the protein. So this comes from a paper of Kobe's where he has noticed that if you look at true clients of the proteasome that are ubiquitinated and known to be clients, many of them don't actually have this required unstructured region in their structure. There's no obvious unstructured region. So the question is, how do they get engaged and degraded by the proteasome? And there's an intriguing hypothesis that was even brought up in the very beginning of understanding the end-to-end role, is that perhaps ubiquitination itself modifies the landscape of the protein such that it's easier to be degraded and engaged by the proteasome. So that's the question we wanted to ask experimentally, and actually Kobe has shown that that is likely, that could well be true by his computational studies. And Andy Martin, who has a lab next door to me, is a world expert on the proteasome. He'd solved the structure of the proteasome, 26S proteasome, and I would always ask him, what the heck does ubiquitin do? What about the client protein? And he said, nobody really knows the answer, because getting the substrate, ubiquitinated, purified biophysical quantities, and studying that is a technically difficult thing to do. So Emma Carroll in my lab took this on as a project, and I'm summarizing, Emma, I'm summarizing many years of work in this one slide. So first, how could she make a system where she had a specific protein of interest? She had only one lysine in the protein. She could control where that lysine was, and she could generate precise monoubiquitinated proteins with an isopeptide true linkage attaching that ubiquitin to the protein. So she found a couple of proteins that would be tolerant to having a single lysine substitution, and all the rest are arginine. In this case, I'm going to show you work from Bar Star. And she used, in collaboration with Andy's lab, she used their recombinant assay. I'll tell you how she makes the substrates quickly. She uses recombinant enzymes from Andy's lab to create a polyubiquitin chain. She also attaches a fluorescein to a cysteine on the protein so she can follow it in gel-based studies. She has attached onto this construct other features, a His6 MVP and a PPY sequence that helps right after and then a GS linker, and this will help the E3 ligase recognize the substrate and efficiently ubiquitinate it. Once she has this polyubiquitinated, she treats it with a deubiquitinase that will chew down the polyubiquitins to the single monoubiquitin. Then she can cut it right here, and voila, she can purify the substrate she wants. So she's got this quite robust. It works better than we ever imagined, and she was able in the end to get enough substrate that we don't even need to use a gel. We can measure its stability through traditional methods. Luckily for us, ubiquitin doesn't have any tryptophan, so we can just do a tryptophan denaturation. And what you see here is that there is a change in the energy landscape or the stability of when we have a lysine at 60 in bar star. So this is the denaturation of unmodified bar star, and this is the monoubiquitinated bar star, and there's about a 2 kcal per mole destabilization. So the answer number one is ubiquitin can destabilize the protein and can energetically affect how the client protein behaves. We then move the lysine around to see if it made a difference to where it is, and we're looking at 3 different natural lysine positions in the protein. And what you can see that these are very site specific. At positions K2 and K60, one is in a helix and one is in a sheet. These are both destabilizing. And because the M values slightly shift, I'm just reporting these in CMs because the 2-state approximation, we could question it, but it's about 2 kcal, sorry. Yeah, I'll have to remember in a second how many kcals per mole that is. But here you see when I put the lysine bar star K78, a ubiquitin on that, there's very little effect. So in other words, the effect of ubiquitination depends on the actual position of that lysine, varying from nothing to something that seems to be quite significant. Now, of course, the global stability of the protein isn't likely to be what's important for the proteasome because, as I said, all I need is a region of the proteasome, of the protein to be disordered in order to engage in the proteasome for degradation. So it's much more likely that a more frequent partial unfolding reaction would be what engages the thing into the client into the proteasome. So we decided to use some pulse proteolysis or proteolysis assays to look for whether or not there were these subglobal unfoldings on these proteins. And again, we turn to proteolysis, as I said, and in this case, we're just measuring, we're using the rate of proteolysis as a probe. So this is very much like hydrogen exchange. If you assume that the native protein doesn't have a fluctuation that's cleavable and that you fluctuate to a cleavable, partially unfolded form, and that this is an EX2, which it is in our condition, that this then cleavable form gets digested. And if we measure the digestion of these cleavable forms, we're going to be getting some information about the free energy or equilibrium constant to this cleavable state. So simply by measuring the rate of kinetics as a function of the protease, we can then extract out the free energy of proteolysis, which is going to tell us about a partial unfolding reaction. And so that's what we did. So here it is for all 3 versions. These are the gels just because we like to show you them, that you can see that the unmodified is digested quite slowly. This is time, whereas at the modified ubiquitinated gets degrades much faster. We've quantitated this here, and you can see that the lifetime changes or the degradation rate changes quite dramatically under these conditions here for bar star K60 and for bar star K2, the 2 same sites that had a change in their global energetics. We can calculate those free energies, and it's about 1 kcal per mole. Now, when we take the non-sensitive site where we don't see a change in the stability, we again see no change in this delta G proteolysis, as though it also doesn't fluctuate to a partial unfolded state of the protein. So these are all in vitro fluctuations. We wanted to know whether or not this energy landscape actually had an effect on proteasome degradation. So we took, before I go there, I know I'm getting quick here on time, sorry. Before I go there, I usually get this question. We've done this on other proteins, and I can tell you that the rate of proteolysis and the effects do not seem to correlate with the size. Here we have SARC-SH3 with a single lysine, and it's very small, yet it seems to be robust to ubiquitination at this site. And here we have DHFR from M. smegmatis, and it shows a dramatic change. In fact, so dramatic that we can't even measure it. Once it's very resistant to proteolysis, and once we add the ubiquitin, it gets degraded right away. It's not as though it's not folded once we add the ubiquitination, because we can measure its binding to methotrexate and see that it does bind there. So the protein is folded but very destabilized. Okay, so let's look at the proteasome degradation. I'll try to get to the end here pretty quick. The first thing we did.
5 minutes or so?
Sorry?
Can you finish in 5 minutes or so?
Oh, for sure, for sure.
Okay.
So the first thing we did was to attach, is to provide from Andy Martin's lab a ubiquitin independent delivery system to the proteasome. So in this case, all of our variants, ubiquitinated or not, will be brought to the proteasome, and we can just see the degradation, engagement and degradation rate of these individual variants, and ask if there's a difference between them and if that difference correlates with the change in energetics. And that's exactly what you see. The destabilized variants are more rapidly degraded than the non-destabilized variant. The non-destabilized variant is comparable to when you don't have a ubiquitin there at all. So we know that these effects of ubiquitination are site-specific, and then they can affect the ability of the proteasome to degrade these substrates. So we wondered if actually ubiquitination alone could provide a disordered segment that then could be brought to the proteasome and degraded. In other words, could we create a bona fide client for the proteasome by adding ubiquitin? So that's exactly what we found. If we add polyubiquitin to these proteins and watch them be delivered the normal way to the proteasome and degraded, what you can see here is we see degradation when we put bar star K60, which is the sensitive one, bar star K2. But then when we do the non-sensitive one, we don't see degradation. This is just consistent with deubiquitination slowly over time. Moreover, this is definitely related to the energetics of the protein, because we can stabilize the protein by adding baranase, its partner, and this degradation goes back up, similar to what it is for a non-client. So we can regulate, these changes are definitely there. So putting this all together, what you see is that there are ubiquitin-dependent changes in the energy landscape. It depends on where you put the ubiquitin, and that we can modulate that these changes have phenotypic consequential changes for the degradation, suggesting that ubiquitin itself provides a method of regulation of who's a client for the protein, for the proteasome, and who isn't. And this can be, we've shown, which I haven't shown you, is things like stabilizing mutations, ligand binding, and deubiquitination. So just in the last slide is a way to show you where we're going with this. What is the molecular mechanism? How is ubiquitination doing this? And we've taken many different prongs. We're just wrapping this up now. So I just want to give you a hint. First of all, the structure, as has been seen by others, the structures don't seem to be changed by ubiquitination. The HSQC spectra are all the same. There's no obvious differences in the contact densities of these different sites when we look at it computationally. And we don't see any interactions between the ubiquitin and bar star. And Naomi in my lab has been doing molecular dynamic simulations. We do see some differences, however, once we ubiquitinate in the fluctuations from the native state as monitored experimentally and computationally. So what I'm showing here actually is, first, if we do MD simulations, we see that the regions that are more sensitive become more dynamic. But here I'm showing you an experiment where we've done HDXMS. We're just looking at the protection or the different residues, different peptides in the protein to look where we might see fluctuations. And I'm plotting here the differences in the hydrogen exchange rates plus and minus ubiquitin. And what you can see is there are very little changes. Red means they become less protected upon ubiquitination. Blue means more protected. And what you can see is the C terminus becomes more flexible in both of the sensitive variants, those that are clients for the proteasome. And we don't see any obvious changes in the non-sensitive one, but I have to remind you, we can't actually look at the C terminus here because it is at the C terminus, and the scar of the ubiquitin causes problems. But all of our computation and other work suggests that this is a allosteric effect where the ubiquitination causes partial unfolding at the C terminus, which I didn't tell you is likely the part that engages into the proteasome. Sorry, that went a bit over. Okay, so in summary, The ribosome, I've told you about the birth and death. The ribosome generally appears to destabilize nascent chains. Co-translational folding can reroute the folding trajectory, avoiding intermediates that may be prone to misfolding. Ubiquitination can affect a protein's energy landscape. It has important effects for proteasomal degradation. And this is a new level of potential regulation for proteasome clearance of a substrate. So lots of people have helped with this. I think I mentioned them along the way. This is really a large group effort. We have lots of collaborators. Here are some of them. And I, again, think I mentioned them along the way. And I want to thank all of you for keeping our community going. And just, I miss you all terribly. And thank you for giving me this opportunity to chat.
Thank you very much, Susan. So as I mentioned in the beginning, a breathtaking odyssey of techniques and beautiful results. And I'm sure there will be lots and lots of questions here. So we are going to start with Stephen Fried.
Wonderful, thanks. Can you hear me?
Yes, hi, Steve.
Hey, no, wonderful talk. Thank you so much for joining us, even at a crazy time and under a crazy set of circumstances. I loved, specifically, really loved the HaloTag story. And I'm sort of like looking at it anew from sort of like fresh eyes, because when you showed the structure of that protein, I'm trying to remember, but is it basically like a situation where you have like a sort of split domain where like one domain is lying inside another domain? It sort of looked like that from the way that you drew this.
Yeah, a little bit like a discontinuous domain. Is that what you mean?
Yeah, exactly.
Yes. So it is somewhat, if you squint and blur and be a PI and look at it not very detailed, that is what you see. The bottom, there's a lid, and then there's a bottom part of it. And so it does seem to be that way. And that, as you know, is why Natalie's trying to look at these circular permutants.
Right. So one thing that you might find interesting is that when we recently sort of did a lot more like a domain level analyses on our like refoldability data. And we found that proteins that have that topology where one domain is basically split by an internal domain are essentially never refoldable. They're like the least refoldable category of anything that we've seen so far. And our sort of hypothesis for that is that basically when you have like these like kind of like two flanking domains that have to sort of, or two like I guess regions of a domain that have to sort of like reassemble, like that seems to be something that's very hard to do in vitro, and maybe something that the ribosome is basically helping, that's kind of allowing for that internal domain to sort of fold on its own before you have like the C-terminal region of the first domain that's trying to go back. And I was wondering, would that be consistent with your HDX data? Because you sort of showed like there was a region in red that was like to the flanks and a region in white that was in the center when you were showing.
Right, so that's the refolding trajectory. We don't know what the co-translational folding trajectory is, but of course, Judith Friedman has also shown, you know, a long time ago that multi-domain, the ribosome acts as a whole base to allow one domain to fold before the other, and you not get screwed up along the way. So that model is very attractive and likely, and fits with our data as well.
Okay, cool.
But I don't know what the intermediate that does form co-translationally. We still have to find that out.
Right. Well, I guess the hypothesis that I would maybe offer would be one in which like the internal domain basically completes on the ribosome, that like completes its folding, which then basically makes it easier for the C-terminal part of the first domain then find its partner off the ribosome. Whereas if the whole process is happening off the ribosome, then maybe it just gets confused because it has like too many options. Yeah.
So for sure, something has to happen on the ribosome. Otherwise, we wouldn't see the effects we do. Something must happen early. Exactly.
Thank you, Stephen. Next question is Ben Schuler.
Thank you very much, Susan. A really very elegant combination of ways of addressing these very complicated questions. I have a question regarding the somewhat surprising lack of an effect of ubiquitination at lysine 78 compared to the others, just the equilibrium stability without any proteasome or whatever. Is there any obvious reason if you just look at the possible interactions or sites that are involved?
So we're looking at that now. Naomi's doing a lot of molecular dynamics simulations, and we do, as I said, and we've done NMR HDX in the absence of ubiquitin, where we can look at it residue by residue. So, we have 3 sites. We're making a lot of conclusions on 3 sites. And the 2 sites where we do, it's clearly not secondary structure. And Kobe had put a nice suggestion out there that it was contact density. We don't see differences there. We do see that they are more dynamic sites, the ones that are less rigid, if you would like, in the apoprotein and just the protein alone. So whether or not that has something to do with it, to let it explore different things, I think it's clear there's not going to be a single mechanism for destabilization. It's going to be different at each place. We do see something which I didn't have time to, which is kind of cool. I don't know if it's related or not, but we see in the snapshots in the MD simulation where the ubiquitin substitutes for that strand in the K78 situation where we form a protective sort of different hydrogen bonded network. Whether or not that is a real thing or not, I don't know. We could go and look at, we also don't see any effect on SH3 when we put a ubiquitin on it. So I don't know what's going on. As you can imagine, we're also trying to pull on these proteins to see how they mechanically unfold. So that is the question we're trying to work at now. I don't have that answer for you.
Thank you.
And if that's unique, K78, or if it's going to be a common thing, I don't know.
Yeah, thanks.
Okay, Shahar?
Yep, thanks. Thanks a lot, Susan. That was a fantastic talk. So I was wondering, first of all, is there like a question of like chicken and egg now? If the ubiquitin itself destabilizes the protein, is that just sufficient to cause degradation? And maybe the flip side of that question, did you try doing the same experiments on stuff that's otherwise misfolded or, destabilized to see if the effect is stronger or kind of the same?
So to answer your first question, we've definitely shown, at least for this bacterial protein that's not a client of the proteasome, that adding the ubiquitination is sufficient to make it a client. It doesn't get degraded without it. It does with it. So I think it can, technically it can do that, whether or not, it is the mechanism in the cell. There are obviously other unfoldases that are known to contribute to present the protein to the proteasome. Mm-hmm. So I think we can say that it can, and it is a very attractive and potential mode of regulation. And that's why we're excited about it. I'm not sure I understood your other question, but we haven't done it in terms of misfolded. We have taken Barstard and introduced stabilizing mutations to it, and then added the ubiquitin, and everything scales as you would, in that now it isn't, we can't, the ubiquitin is not enough to do it because it's too high a stability. So the actual stability of the protein and its fluctuations are going to matter. Does that make sense?
Mm-hmm. Yeah, it does. Thanks a lot.
I hope the air quality is a little better where you are today. It looks better.
Today it's fine. Yeah.
Yeah. Today we have blue sky.
Okay. Next question, Yuri Lubchenko.
Susan, it's really great talk. The question that I have, it's actually relating to your data on a strong effect of ribosome on protein folding. I look at another perspective to this data. Could it be that they suggest the role of ribosome surface as a local environment, which somehow contributes to this effect? If this is true, how about other surfaces? For example, our data, they revealed the effect of membrane surfaces in relation to protein folding, misfolding, dramatically increasing aggregation process. What do you think and what happens with ribosome?
So if I understand your question correctly, you're asking how much of this is just being tethered to a big surface, and is that the effect that we're seeing? And we don't know the answer to that. And clearly there's lots of evidence, as you point out, that just putting something on a charged surface is going to affect things. Are there specific effects between ribosomal proteins and the nascent chain? And there's, that's really what John is looking at quite nicely, and with his NMR, whether or not that's general or specific for specific proteins. I think those are really great questions that we hope to answer in the future. We don't know. We do know that if we do this experiment by adding ribosome and adding protein and they're not translating, right, we don't see this effect. So it's not just being in the presence of the ribosome that does this. So I don't have any other answers to that.
Next question, Kobe.
Hi, Susan. Yeah, so it's really, really fun to hear these results. I of course read your paper. It's the first time that I hear it from you in the lecture. So these Zoom seminars are really useful for me. My question is, there are several examples that indeed monoubiquitin is related to degradation. But usually it is believed that you need these larger polymers of ubiquitin with a specific connectivity. So there is kind of many types of polymer of ubiquitin. And each of them usually is associated with different functions. And I believe that each of them should correspond to different degree of destabilization. And probably these larger polymer of ubiquitin will result in larger destabilization than the monoubiquitin. So I wonder if you have any plan to work with different types of polymers of ubiquitin, or maybe you already have some results.
Yeah, no, great question. So as you all know, Kobe really generated all of that work with his great papers stimulating us to think about this. As you know, we were working with K63 polymers. And when we looked at polyubiquitination versus monoubiquitination, that we didn't see in our gross proteolysis assay a big difference. That doesn't mean, it was a heterogeneous mixture. Technically, I don't know, but of course, Emma and my students may know better, that haven't figured out a reasonable way to do exactly what you're saying. We were lucky to have the reagents to make what we did from Andy Martin's lab, to ask if we present, a different tetraubiquitination that has a different surface, whether we would see different effects. But I think obviously that's what I would love to be able to figure out to do in the future, and see if there is any difference. And hopefully Emma just graduated, I can convince somebody else to come on and do those projects.
Okay. Any other questions? Chu Tang.
Hi, Susan, I have a question. So, you showed the bar star has two sides of ubiquitination have effects on the stability. So what about other kinds of modifications, say acetylation on the same side, does also affect the stability of the protein?
So, there's lots of things: pegylation, acetylation, phosphorylation, and I personally haven't studied any other post-translational modifications, but one would expect, they have, I think people have studied some of those, glycosylation, et cetera. Ubiquitination is unusual to us because it's very large. And so there's been, Jane Clark, lots of people have studied that multi-domain proteins, including polyubiquitination, that the different domains will have different stabilities. So that's all we know. We haven't looked at any others, but I think that's something we all have to look at, whether or not these are, phosphorylation is a charge. It's obviously, these are mutations. So we do believe they are, but I don't have any data to answer that.
So I have a question which might be a bit specific. So when you showed the HaloTag folding, you had a lag time in the folding in the beginning before it started to go off. Okay, do you understand what is the reason for that lag time?
So what we do is we put DNA and enzymes in a test tube or in a cuvette. And so the DNA has to be made into RNA that then has to be translated. So that lag, that lag time is going to be that. And that's modeled into Ed O'Brien's modeling of it. Which is which, I guess I could figure out by instead adding RNA or something like that. But yeah, it's not unexpected for sure.
Okay. Okay, so it looks like there are no further questions. So I would like to thank you again for this great talk. And thank you all for being here, and hope to see you in 2 weeks again in the talk of Henry Chapman. Thank you very much. Bye-bye.
Thanks.